Hello everyone. So welcome to the 8th day of Thursday for Theoretical Biology 2021. So we have two speakers for the day. So the first to start off will be Professor Vidyanand Nanjundiah. Vidya is a long-time faculty at the Indian Institute of Science. He's a trained physicist, but moved to biology very early on. After IISc, he has been at the Center for Human Genetics and also at the ICTS from a couple of years. So Vidya will speak to us today about Turing's the chemical basis of morphogenesis. Vidya.
Thank you, Vijay. Welcome everyone to this meeting. Today's session will have two speakers. Professor Shigeru Kondo will follow me. I may not take the full first session, but we'll split it with Professor Kondo, who will take over the second session by himself. So you see the title of what I'm going to say there. Whoops, the screen isn't moving. So my talk is going to deal with this paper, essentially what led up to the paper, a little bit of what was in the paper, and a few comments towards the end on what followed later. So you can see the outline is really very simple. Before Turing, by which I mean the Turing paper, which you saw a moment ago, post-Turing and beyond Turing. And the meanings of these terms will be apparent as we go on. So before Turing, the general theme of this paper of Turing's that we are discussing today is development, multicellular development if you wish. And this is a standard cartoon of the course of multicellular development in a frog, in a toad actually, called Xenopus. The time scale is indicated there. The spatial scale is about a millimeter or thereabouts. So the egg is about a millimeter in diameter. And these stages are very familiar to us. Cell growth due to metabolism, cell division, movement, the formation of specific cell-cell contacts, intracellular signaling between cells, and then cell type specific gene expression. All of these things are going on during development. And the particular aspect of development that we're going to look at, namely pattern formation, which is obviously related to the spatial distribution of cell types in the embryo, pattern formation involves all of these steps. A few landmark findings or results in the history of developmental biology to get us going. Roux showed in 1888 that if he took an early embryo of the frog again at the first cleavage stage and killed one of the two daughter cells by using a hot needle essentially, you have to kill the nucleus, so you have to poke the needle in to some extent. That cell died, nothing further developed from it, but from the cell which was remaining untouched, what you got was the development, what he got was the development of a half embryo, in fact, precisely that half of the embryo that would have developed in the normal course of events. And this was taken as an indication that already at the first cleavage stage, the embryo was differentiated in terms of its potencies between a left half and a right half, each of those halves being capable of fulfilling its normal role in development, whether or not the other half was present. And by an extension of ideas based not just on this experiment, but on many other experiments, in fact, many other more pointed experiments, the notion grew that perhaps embryonic development involves an already mosaic cytoplasmic distribution in the egg, namely an already differentiated cytoplasm within the egg, so that cells which following cell division came to occupy different regions of cytoplasm would be potentiated to differentiate in different directions. Now, this sort of thinking has a very old history in developmental biology. It goes under different names. It goes back all the way to the notion of preformation, but mosaicism is the term under which it's generally known. What it says, pushed to the extreme, is that cells develop in some kind of an autonomous fashion with interactions between them being insignificant. However, at about the same time, Hans Driesch, another distinguished embryologist, did a different experiment. In fact, he did this with frog eggs as well as with sea urchins, which you see here. So the sea urchin experiment involved allowing the embryo, the fertilized egg, to divide, in this case, 2 times. Both of these divisions, as it happens, are what you might call vertical divisions at 90 degrees to each other. So you get 4 cells, and it's possible by simple tricks to separate these 4 cells. And Driesch discovered to his surprise that each of these 4 cells was capable of giving rise to a perfectly normally proportioned larva, what's called a Pluteus in this case, which then went on to give rise to an adult sea urchin. The implication being exactly opposite to the one that was drawn from Rooks's experiment, namely that the cells following these initial divisions, at least the initial 2 divisions, were totipotent. During normal development, they did not express their full potency, but were somehow constrained by the presence of other cells to develop into, in this case, roughly speaking, the anterior left half, the anterior right half, the posterior left half, and the posterior right half. This sort of development, where the fate of a cell seemed to be not fixed forever, but was labile, came to be known as regulative development. Roughly speaking, regulative development implies, or seems to imply, that a part of an embryo is equivalent functionally to a whole, except that it's not permitted to express its full potentiality under normal circumstances. And the conclusion you would draw from this kind of embryonic development is that development is not autonomous, the course of cellular differentiation is not autonomous, but that it depends on the social context, if you wish, or depends on intercellular interactions between cells. The third crucial experiment in this sequence, which came quite a while later, you've heard of this from Erez Brown during his talk, was the organizer experiment of Spemann and Mangold. As he pointed out, they were preceded, they were preceded in this work, in this discovery by Ethel Brown, who discovered the same sort of phenomenon in Hydra. So basically the observation here is that you take a piece of embryonic tissue, transplant it from its normal location to a different location. You do this at a very early stage, in fact, just before the onset of what's called gastrulation. And if you do that, this piece of tissue, which is really a rather small square, perhaps no more than 2 or 3% in terms of surface area relative to the whole embryo, this piece of tissue seems to have a magical power to induce the formation of a second embryo. And in fact, you get these so-called Siamese twin larvae, which is from one of Spemann and Mangold's papers. So there are 3 findings I have put across to you. One, seeming autonomy in development, mosaicism. Second, seeming non-autonomy in development, regulation involving intercellular interactions. And thirdly, some regions of the embryo may have special properties. By the way, I've not gone into details here in this experiment. As you can imagine, you have to do the appropriate controls. You have to take out some other piece of tissue, do the same transplantation, see if the result is obtained and so forth, so as to convince yourself that it's a property with this part alone. So a number of conceptual constructs came up in the late 1800s and early 1900s, And these had to do with the terms determination and differentiation and the distinction between the two terms. The point being that, as you saw in this transplantation experiment of Spemann, cells seemed to have hidden potencies which were not yet expressed at the time that you looked at them, but those potencies became expressed later. In fact, there were a number of experiments along similar lines, not quite as dramatic as the Spemann-Mangold experiment, where a piece of tissue, if removed at one time, would not result in any damage to the embryo, but if removed at a later time, would result in irreversible damage. In fact, would result in the absence of whatever it was in the adult body, that tissue would have gone on to differentiate. So experiments of this kind made people think in terms of these two concepts, one being determination and the other being differentiation. The idea being that there's something going on inside cells and tissues before you see the result of what's going on. And in terms of theories or models, it allows for an effective separation of timescales. The second point, regulative vis-a-vis mosaic development, we've seen already. The third point was that a great many embryos seem to have a well-defined axis of development. So the frog embryo that you saw earlier has a very clear so-called animal-vegetal axis, which mirrors the future head-to-tail axis of the tadpole larva. So the embryo was anisotropic. In addition, a number of experiments, and you saw these illustrated again very nicely in Erez-Brown's talk in the context of Hydra, a number of experiments showed that in some cases at least, there seemed to be evidence for graded differences in differentiation potencies in these determinative capacities that I mentioned a while earlier. And these graded differences indicate that there were inhomogeneities within the embryo, quite often within an axis which you could identify and study further. So these are the four theoretical concepts which come into the discussion that proceeds. One is an effective separation of time scales. The second is autonomous versus non-autonomous behavior. As you will see, we will concern ourselves only with non-autonomous behavior. The third concept is that of anisotropy, and the fourth is inhomogeneity. Now, this slide has a lot of detail, which is simply an expansion of what I've told you so far. I'm not going to go through the slide at all. The take-home lesson from this is that there is this separation of time scales that I told you about, namely cells and tissues commit themselves to doing something before they do it. Secondly, this commitment takes of the order of 2 to 3 hours and occurs within group of cells, within groups of cells, which in their linear dimension are about 1 millimeter in size. As we've already seen, regulative embryos imply intercellular communication, and this communication is assumed in the situations that I'm going to look at, assumed to take place via molecules, either small molecules or large molecules, which are in any case gene products, primary gene products if they happen to be proteins, or in the cases that we will look at, secondary gene products, which are products of enzyme action on various substrates. An important assumption in what I'm going to present before you, because this is what Turing made, even though he realized that this was a limitation on his models, is that growth, cell division, and mechanical forces could be ignored, so that at least in terms of this phase of determination, of commitment, you could think of a tissue, 100 cells roughly, which is about 1 millimeter in any one direction, within which some sort of imprint was laid down of the future differentiation which could occur within this tissue, which you would see much later. And for this imprint to be laid down, it took 2 to 3 hours. Remember, we're in the before Turing phase. So a couple of other conceptual antecedents to Turing, which I will not go into much more. Again, this is the name that you've heard, Darcy Thompson, who came up with a theory of form, which was very different from that of Turing. It was an evolutionary theory that Darcy Thompson presented, but based strongly on mechanical forces. And this is the famous picture from his book on growth and form, one of a series of pictures where he showed that two fishes that came from entirely different groups, so they were not even the same species or the same genus, could nonetheless be shown to resemble each other in outward shape by an appropriate mechanical transformation. I won't go into details of what Thompson said here, it doesn't concern us, but please keep in mind that he laid his emphasis on mechanical forces, precisely what we are ignoring now, or going to ignore now. Right. Something else which comes here in this before Turing phase, even though it did not concern Turing himself directly, is this business of what's called lateral inhibition, illustrated here by the formation of what are known as Mach bands. So what you see here is a left gray half and the right darker half in the rectangle on the top. The luminance gradation between these two halves is as shown in the middle rectangle. So flat at the left, flat at the right, and a smooth gradient in between. The perceived luminance, however, has a peak at the two boundaries, a positive peak on the left-hand boundary, which looks whiter than the rest of the white to the left, and a negative peak on the left, on the right boundary, where you have a stripe which looks blacker than the rest. And this, as we know, thanks to the work of Hartline and others, is due to what's called lateral inhibition, essentially a suppression of the excitation of visual retinal cells by neighboring cells. Please keep that in mind. Right. One more piece of background to what I'm going to say, except that I won't go into this at all, quite obviously, but those who are interested in differential equations can take a look later. It turns out that quite a few people, in fact, used mathematical apparatus which resembles the sorts of things that Turing did, which we will look at in a moment, except that they used these in contexts which were very different from development. So the Lotka-Volterra model used in an ecological context. Rashevsky did use it for development, but then he also used it in many other situations, including nerve excitation. Fisher and Kolmogorov used it again in an either ecological or population genetics context. And the Hodgkin-Huxley equations, of course, were developed for explaining the spread of nerve excitation. All of these equations can be shown to resemble, in a formal sense, the Turing equations, which is to say can be shown to resemble a diffusion equation with sources or sinks. Please keep that in mind. It's interesting that none of them were really used by Turing in his thinking. It's only retrospectively that people have come back and said, look, there were these antecedents to Turing after all. Right. Now, Turing himself, what does he say in the paper? Well, first of all, that's Turing. He had many interests, including long distance running. Turing in this paper wanted to do a number of things, wanted to explain anisotropies arising on the surface of the embryo leading to gastrulation, wanted to explain symmetric structures in development, for example, starfish, or the arrangement of flowers, arrangement of the flower rudiments along the stem, leaf arrangements. In fact, there's something interesting about the Fibonacci series that he worked with, which people are discussing to this day. Also wanted to explain color patterns on animals, you know, the stripes on the back of a zebra or the spots on the back of a giraffe, so on and so forth. Dappling patterns, so the sort of seemingly random patterns that are quite commonly seen on many animals, including cattle, and patterns or symmetries seen in structures that have more of a spherical appearance, such as radiolaria. And in this letter, he says it's doubtful that I can achieve that, but he wanted to do that. So this is the Turing system, not his own equations. This is copied from a place which has written the equations in a form that makes them understandable. You have two variables, u and v. The left-hand side of each of these equations expresses the rate at which these variables change in time locally. And the right-hand side describes the factors which go into these concentrations changing. And the factors are roughly chemistry, described here as production, degradation, which is a kind of default term which says things decay by themselves, and diffusion. The chemistry term turns out to be central to Turing's analysis because it's the right kind of chemistry, the right combination of positive and negative feedbacks, which give him the patterns that he's looking at. And the way the system works is rather easy to explain. So you have these two components, remember, U and V, and the way they interact, and I'm going to come to that a bit later. So the way U and V interact, which goes into these capital F and capital G terms, is the following. Try and imagine that the initial picture that you see there is a small blip in the concentration of U as a function of space, as a function of distance along the line. Assume that the concentration is uniform everywhere except for this blip. Now U has the property that it can positively stimulate the synthesis of V, beg your pardon, which is shown as a dotted line. So B shows the second component V picking up as a result of the stimulation by U. Now it's assumed that V can diffuse more rapidly than U, so it has a diffusion coefficient higher than that of U. Therefore, if you move some distance away from this blip, and you see this already in B, the concentration of the second component V is higher than that of the first component, as a result of which the second component, which in contrast to the first component has a negative effect, depresses the concentration of the first component. You see that in C. And having depressed it, it turns out in some models this varies, it can also pull itself down. So you finally end up with a pattern such as D with peaks of both U and V, peaks of both the positively influencing component and the negative influencing component, followed by troughs. And both this pattern and the initial blips that you saw at the beginning are due to fluctuations alone, meaning that in these equations that you saw a while ago, you begin with the assumption that nothing is changing, that everything is uniform. And these fluctuations allow you to set up this blip, which is an anisotropy. Now you can show, if you go into the equations, that these blips can occur in various places all the time, but their effectiveness is limited by the distance to which V and U have spread from the first peak, which you see here. Therefore, this gives rise to what's sometimes called a chemical wavelength, which is a distance between two such peaks. And finally, you get up a pattern of peaks. This is one of a number of results that are available. The discretized version of the equations that Turing actually worked with are shown on the top. And the outputs that he got varied from case to case. Sometimes he got stationary waves of extremely long wavelength. Sometimes he got oscillations, which were synchronized throughout the field, what you would call bulk oscillations. Sometimes he got stationary waves of extremely short wavelength. In his discretized model, this meant that neighboring cells were doing things that were the opposite of each other, were in opposite phase to each other. And finally, you could get stationary waves of intermediate wavelength, let's say finite wavelength. So you got a whole range of outputs possible. One simulation that he actually did, numerical simulation that he actually did with numbers, was to try and replicate in some sense the tentacle pattern around the mouth of Hydra. And as you can see on the left, this works out in some rough fashion. He takes a ring of cells here, the equations that you saw a moment ago, and bunks them in his computer and gets these evenly spaced peaks around the ring. Something else that he worked out was this sort of a dappled pattern, and this was a two-dimensional pattern, as you see, and this in some way approximates the kinds of things that you see on the backs of. Okay, so I've done with the first two parts, and I'm going to pause briefly here in case there are any questions. Vijay, are there any?
No. So far, no questions with you.
None at all. So shall I proceed?
Yeah, please.
Please.
Okay. Now, let me just briefly summarize where we are. So Turing was interested in addressing the problem of how biological patterns developed. He focused his attention on rather simple patterns. The approach he took was to say that there were chemicals which could move from cell to cell. Their movement could be approximated by diffusion. Within each cell, these chemicals could interact with each other. This combination of movement and interaction was sufficient to give rise to non-uniform distributions of these chemicals in space or in time or in both. This basically was Turing's finding, and the reason it created a lot of interest, at least among people who were interested in modeling, was that this seemed to show really for the first time that you could get structure, form appearing in a biological system without putting anything into it. Because as I said, his model essentially said that you begin with a uniform steady state and spontaneous fluctuations combined with chemistry and diffusion are enough to give rise to structured pattern. In contrast, there were a number of views, some of which went along with Turing, some of which pointed in different directions. Many of these views preceded Turing, but they were firmed up, at least in print, after Turing. So we will call this the post-Turing period. Curt Stern, who you can make out from this picture, had genetics and chromosomal dynamics as a main interest, spent a lot of time trying to use genetics-based experiments to show that the kind of thing that Turing had done, even though he doesn't explicitly say that's what he's doing, Curt Stern used genetic experiments to show that the kind of thing Turing had done could actually be accommodated within the framework of genetic analysis. And Stern did this by carrying out a number of experiments where he said that before a pattern develops, you have something that he called a pre-pattern, which is cryptic. You don't see anything, of course, but it's there. And it's this pre-pattern which arises spontaneously, which turns out to be the basis of the future pattern. Really pretty similar to what we've been saying all along and what Turing said as well. The interesting thing about Stern's study, as I said, Stern's approach, as I said, was that he said this pre-pattern was based on genetic influences. So he did a lot of work isolating mutations which were deficient in the pre-pattern in one way or the other. And he's written a very fine book, it's really a collection of essays by him called Genetic Mosaics, and I strongly recommend it for reading. However, the experimental evidence that I want to show you comes not from Stern himself, but from Maynard Smith and Sondhi, who actually applied these ideas of Stern to see whether this pre-pattern kind of idea, please remember, it's essentially the same idea as that of Turing's, except that it explicitly invokes a genetic underpinning. So Maynard Smith and Sondhi carried out a number of experiments to see whether this pre-pattern kind of idea could be usefully applied to explain the distribution of bristles, of hairs on the fly surface. So these are pictures that you see at the bottom from the fourth abdominal sternite, which is the ventral body segment, abdominal body segment of the fly. And you see the real bristle patterns shown there. And what you see on the top are cartoon patterns. These are Turing patterns based on where the peaks and troughs of the distributions of those chemicals that we were talking about a while earlier, U and V, would come. As you can see, the cartoons do not agree precisely with the observed patterns, but with a little bit of fiddling, in other words, a little bit of parameter variation, Maynard Smith and Sondhi were able to show that you could actually get pretty good agreement. More interestingly, in line with what Stern had advocated, they focused on mutants in Drosophila, which were already known, where the bristle pattern was affected in specific ways. So you had aberrant patterns because of the mutation. And they were able to show that they could mimic these mutant patterns by now doing something with the parameters of the model, of the reaction diffusion scheme, which means either fiddling with the diffusion coefficients, or fiddling with the chemical reaction rates, or fiddling with the feedback components. In any case, they set up a persuasive case, at least in the for the Drosophila bristle pattern situation. They worked with other bristle distributions as well. So, for example, on the head, they set up a persuasive case for saying that this Turing kind of explanation and a pre-pattern kind of explanation was really quite plausible. There's a reference to a review paper by Sondhi, who, by the way, died tragically early on account of an automobile accident. That review by Sondhi is really a classic. It is worth reading by anyone interested in patterning. Somewhat later, in 1969, Louis Wolpert came up with the concept of positional information, which has had an interesting run over the past 50 plus years in the following sense. Wolpert set up the concept to try and explain what he thought was a universal feature behind different patterns. Now, this word universal is to be used within inverted commas, if you wish. He specifically meant it to apply to regulative patterns, therefore, the kinds of patterns that we are interested in. And he also said that behind this concept of positional information was the following idea of his, which he developed from the observation of so-called universality. And the idea was that cells differentiated according to their positions in a field, measured relative to the boundaries of that developing field. Therefore, it was not position per se, but relative position that counted. And the grid coordinates specified by this field, which was most of the time two-dimensional, the grid coordinates specified by this field were universal, which is to say, you could be thinking of a chicken embryo or a frog embryo at comparable stages. That's obviously an important consideration. In both of these embryos, a cell would know that it was, let us say, 30 degrees north and 50 degrees east. That would constitute a location-specific signature for the cell, and the cell would have instructions within it, within its genome, which would tell it, if you are at 30 degrees north and 20 degrees east, you differentiate into such and such. Now, what you differentiate into, of course, depends on whether it's the chick embryo that you're looking at or the frog embryo that's looking at. So Wolpert had this idea of universal patterning mechanisms at the stage of geometrical location, at the stage of specifying geometrical location. Now, I'm not going to go into this idea any further, except to say that to my mind, there is to this day no clear validation of the concept of positional information as Wolpert originally used it. However, the phrase positional information has caught on like wildfire, and it's used most of the time to mean that cells differentiate according to their positions, which of course no one would disagree with. I leave that for the moment. The interesting thing is that Wolpert started out saying that his concept of positional information really had parallels with the concept of pre-pattern. That's the top quote. But he came to realize soon, and many people saw this at around that time as well, that in fact these were two radically different concepts. So the positional information concept really said something about the boundaries specifying relative position and this universal location algorithm read out by the cell, whereas the pre-pattern concept said that cells keep talking to each other all the time and decide on whatever information they get locally. Just very quickly, the sorts of experiments which Wolpert used to infer what he called, what he thought of could be universal. So these are the leg of a fly, that's the thing that you see at the bottom, and the head of the fly, which you see at the top, the head plus antenna, containing the structures appropriate to these segments. And the sort of observation which fueled Wolpert's thinking was, for example, that when you had mutations in which, as happens in flies, these are called homeotic mutations, mutations in which you have a part of the head replaced by a part of the eye, the part which is replaced has a homology in terms of position with the part that does the replacing. So for example, if the mutation results in the terminal segment of the antenna being converted to leg, it gets converted to the terminal segment of the leg. If it turns out to be a more anterior segment of the antenna which gets converted, it turns out it is the more anterior segment of the leg which is converted, and so on and so forth. And the arrows in this picture show you that is actually a fairly impressive one-to-one correspondence between axes in these two parts of the fly. In a sense, these two limbs. The evidence went further than this, but I just show this to you to show that Wolpert's thinking was based on solid ideas. Now we come to Girer and Meinhardt, who I think it's fair to say really set the ball rolling as far as discussions of Turing mechanisms and taking Turing mechanisms seriously goes. They worked with equations very similar to that of Turing, but in this case, they did not try to motivate their equations using arguments based on chemistry, which Turing had done. But they did something very nice, which is to formalize what Turing had done and what they did as well in language that made it immediately apparent what was happening. And it turns out that this language was borrowed precisely from the kinds of neurobiological results which I showed you earlier, these Mach bands. And the language used the concept of local activation and long-range inhibition, sometimes also called lateral inhibition. So these U and V chemicals of Turing were replaced by the symbols A and H for activator and inhibitor. The thick arrows show you the feedbacks, red for positive feedback and blue for negative feedback. The thin arrows show you, sorry, I should word that differently. The thick arrows show you self-feedback, positive and negative feedback, whereas the thin arrows show you cross-feedback. So you see that Turing's system of chemicals can be thought to be two chemicals, one of which exerts positive feedback both on itself and the other, that's the activator. The other exerts negative feedback both on itself and the other, that's the inhibitor. And as I mentioned before, in the case of the inhibitor, it's not essential that it exerts negative feedback on itself. But using this scheme and making the Turing scheme less restrictive by bringing in specific sources, so the rho term that you see in the first equation is a source term, Girer and Meinhardt were able to explain not only a great many spatial patterns, but also the results of interesting manipulative experiments like those on Hydra. So these are 3 experiments which show the results of what happens if a segment, an anterior segment of Hydra is cut off. That's the lower left slide. You get regeneration and a restitution of the normal pattern. And what happens when you take just the head and graft it on a previously amputated segment, and now you get interesting results. It turns out that if the graft is made close to where the previous head was, it doesn't take. On the other hand, if the graft is made far away from the previous head, then not only does the graft take, but a secondary head develops. So all sorts of interesting results, which you've heard a little bit of, once again from Eric Brown. And the time at which you make the graft also matters. So all of these things went into their thinking, and they provided detailed comparisons, at least at the level of observations, broad observations between their models and experiments. Still, they had no way of identifying what chemicals they were dealing with. So to that extent, their models remained formal models. Then other things happened in this post-Turing phase. Bard and Lauder did an interesting study in which they pointed out something which Turing himself acknowledges in his paper, that the fact that the paper results in this chemical wavelength, the fact that this scheme, I beg your pardon, results in a fastest growing wavelength, means that you have some kind of a scale limitation in the model. And if you work with a model which is half as big as the one that you worked with before, in other words, you have a line of cells which is cut into two, then there's no guarantee that the previous pattern will be replicated. To put it in technical language, the Turing model is not scale invariant. So it does not regulate in the sense that we wanted it to. If you go back to that sea urchin experiment and try and imagine that what Driesch did there was to take a straight line and break it into 4 parts and get a perfect sea urchin from each of those 4 parts. So this does turn out to be a feature of the Turing model. You'll see later, I think, how this can be handled later from Shigeru. Okay. Now, this Turing scheme has sort of taken on an avatar of its own. So there are many situations in which Turing-like models have been used away from developmental biology. Otto Rössler, in fact, has shown that it's possible in a Turing system with 2 cells and nearest neighbor communication to get quite exotic dynamics, including chaos, which he tries to say in the form of turbulence, chemical turbulence, which is going on in the system. So these are phase plane diagrams of the variables of the system. And the idea is to show that they go through very complicated orbits in phase space, which are not closed, which using mathematical techniques you can show are chaotic orbits. So this is completely different from anything that had been shown so far. Interestingly, by the way, Rössler calls the scheme not a Turing scheme, but Rashevsky-Turing scheme. So as I told you, some people insist on giving Rashevsky his due, which is perfectly all right. But it turns out to be a minority of people who do this, right? Newman and Frisch did something very interesting. On the face of it, this doesn't look like a Turing scheme at all. This looks like a single component which is diffusing. But it turns out, and I'm not going to go into details here, it turns out that in this scheme, which is of course a reaction-diffusion scheme, you can get very interesting patterns on 2-dimensional rectangles, which are the things that you should see on the right-hand side. And these patterns, and again, these are patterns of peaks and troughs, mimic the sorts of patterns that you get, the sort of patterns of cartilage and later skeletal elements that you get in developing limbs. So they did this for a developing chick limb. So there's been a great deal of interest since then in using Turing-like schemes, I should say Turing-like here, because they are essentially reaction-diffusion schemes without the activator-inhibitor input to show limb patterning. It's possible to do a little bit of quite elementary stability analysis to show or derive, if you wish, the formal requirements for a uniform steady state to be unstable in a Turing scheme. And that's done here. Basically, what it amounts to is what we've been talking about all along. which is to say, and that's on the lower part of the slide, which is to say that the diffusion coefficients need to be unequal. Secondly, that the feedback term should be such that if one is a positive feedback, the other is a negative feedback. And the third condition there shows that you could have positive self-feedback and negative self-feedback of these two components as well. In 1986, we are still in the post-Turing phase, a remarkable series of experiments was done by Cox and his co-workers, and this involves the development of whorl patterns in the growing fruiting body of a slime mold called Polyspondylum. You see the slime mold itself on the left. And what this slime mold does, as it forms this fruiting body, which is really like a fungal fruiting body, is to make bunches of cells, spherical bunches of cells, and you see those in the pictures on the right, A, B, and C, detach from the growing stalk as it's growing upwards. And on these detached bunches, you get rather nice spatial condensations of things. Now, what are these things? In their experiments, they were antigens, which these people could label with a particular antibody. All of this happens without any ostensible cell movement or cell mixing. And this spontaneous detachment of these spots that you see beginning to form there results in the appearance, I hope you can see my cursor here, results in the appearance of these outwardly moving elongations, which later give rise to the secondary stems of this creature. So there's a very beautiful example here of model formation. By the way, this fruiting body itself is in itself a space-time plot, because these are the earliest worlds which have separated. More recently, just about to separate, the spheroid has become elongated into an ellipsoid, and this is the tip where nothing has happened yet. The point I want to make is that Cox and his co-workers were able to model this explicitly by a Turing scheme, but then they were able to do something else very nice, which is to show that this Turing scheme, which gives rise to spots, can, under circumstances in this organism, give rise to stripes, which you get where this sphere, instead of being a sphere, is more like a compressed ellipsoid. In other words, in some sense, longish and thinnish structure, in which case the Turing model tells you that what you had as stripes initially, or spots initially, beg your pardon, becomes stripes. So this is very nice, but again, no formal link with real chemist there. Formally. Then, as I said, the Turing scheme has gone on to all kinds of things. The first chemical replication of a Turing model was published in 1990 using a combination of malonic acid, sodium chloride, sulfate, and hydroxide. And you can see that here, under various circumstances, you could get either spots or stripes. The color is because of a color indicator, which is used in the gel within which these things are appearing spontaneously. So this is really spontaneous self-organization of a chemical pattern based on reaction and diffusion alone. And you have two different patterns appearing. Hans Maynard went on to develop his ideas in various fields. And here, for the first time, he incorporated growth. Remember, we had said no growth and no mechanical forces. There are beautiful patterns that he was able to develop for the growing rim of a seashell, which mimics very well, as you can see, the patterns that you see on real seashells. Of course, it needs fiddling with parameters. Much later, Singh and Nusslein-Volhard, and this is work that they've been carrying on, were able to show that the transverse pattern of stripes that you see in zebrafish actually requires a significant component of an entirely different explanation from Turing's, which is a component involving intracellular interactions. So these are the cells which carry pigment granules. They have various names: melanophores, iridophores, xanthophores, and so on. So interactions between these cells plus physical movement of these cells. So the pattern is formed by actual movement of these cells. I won't go into that in any detail at all, because Shigeru Kondo, who is going to come next, will go into all kinds of things, how the basic Turing mechanism is elaborated, what roles mechanical factors might play, perhaps also something about what he has shown, which is the role that can be played by electrical fields and so forth. So the only thing I can say there is, let's wait for the next talk. Now, before I come to the final 2 or 3 slides, I pause here, because in the beyond Turing phase, I'm going to speculate on something, think aloud on something very different. So if there are any comments or questions, please ask me now.
Well, thank you so much for this nice talk. And I believe that we have a couple of questions here. So the first one is, and I apologize already for the way that I am pronouncing your names, but I think that the first one is by Sudipta Sarkar. It's a question about the formation of the pre-pattern. So please, Sudipta, go ahead.
My question is about, sorry, I guess is that there's a lot of feedback. Can you read it out?
Yes, I can read your question if you prefer. So the question by Sudipta is, I did not understand where Turing's equations assume the presence of pre-pattern. The pattern seems to form spontaneously and no pre-existing gradient, doesn't it? Could you please explain?
Turing's explanation did not assume a pre-pattern. Turing's explanation showed where the pre-pattern came from. In other words, Turing was explaining the pre-pattern.
Is that answering your question, Sudipta? I assume it is. Yeah, okay. So the next question is by Sonali. It's actually about the evidence of the mechanism. I am pretty sure that Professor Kondo is going to talk a little bit about that, but please, Sonali, go ahead and ask your question.
Thank you, Javier. Thank you, Doctor, for this wonderful, and it's very new for us. Now, I have a very generic question. Now, the word pattern itself implies repetition, so mathematics could be working. So, is the Turing diffusion model an attempt at a mathematical explanation of patterns, or is there concrete physical evidence or examples of organisms where the Turing diffusion model approximates very well? Thank you.
Okay, I'll answer this briefly because Shigeru is almost certainly going to go into this in detail. To the second part of your question, I'll say yes, there's very good evidence that it applies to real systems, but to see the evidence, wait for Shigeru's talk. For the first part of your question, let me say that what Turing was trying to do was to give what these days we call a proof of principle. He was trying to show that without making any extravagant assumptions, just by using, well, more or less standard chemistry and the mathematics of diffusion, which need not really be the diffusion of stuff, but something which works in the same way that chemical diffusion works, he was able to show that this was enough to spontaneously generate pattern. So I think the thing about the Turing's demonstration that certainly caught biologists was surprised was that you seem to be getting something from nothing. That was the conceptual leap that he made. Then, of course, there were other things which were part of it. One of them being diffusion is always thought to be an agency for smoothing things out, whereas here it seemed to be an agency for creating instability. That, I think, surprised a number of people as well. So these were two features of the Turing model which stand out.
So, sir, can we assume it is emergence of non-randomness from randomness, a mechanism?
If you like, I wouldn't say emergence from randomness, because the statistical factors which give rise to noise. You know, this could be thermal noise, this could be noise due to intercellular signaling, noise due to due to chemical binding to receptors and so forth. So the almost inevitable statistical noise gives rise to pattern. Yeah, you could call it non-randomness resulting from randomness. Yes.
Thank you.
Is there any other questions from the audience? If you do, please raise your hand or write your question in the chat. In the meantime, I will actually raise, I would like to raise a question myself. So many times it's actually difficult to distinguish in biological system if a molecule is actually diffusing or subdiffusing or super diffusing in the environment. So I wonder what could be actually the actual role of the transport mechanism in the induction of patterns if you have not just pure diffusion, but you have molecules that are actually subdiffusing in the environment. Is that changing any way the phenomenology that you have introduced?
I would doubt that very much. In fact, it's easy to show that the Turing mechanism can work even when you have no diffusion at all, no transport of material at all, so long as influences from one cell to the other propagate similarly. And I go back to this example of the Mach bands. So if you have cells which are able to both activate and inhibit their neighbors in the ways indicated with relative strengths of activation and inhibition as shown, then you can get Turing-like patterns. Yes, you can certainly get them. So you don't need anything moving at all.
Okay, so it's just a question about how the interaction is actually distributed in space rather than the mechanism itself.
Exactly.
Okay. I also, I do have another question, because at the very beginning of your talk, you were introducing like some preliminary assumptions, if you want. And one of them was about the role of mechanics in the system. And it has been assumed many times that actually mechanics is not really playing a role in the mechanism, in the Turing stability. But I believe that lately it is more and more clear that mechanics may be playing actually a very important role in the formation of patterns. So can you elaborate a little bit about that, please?
I'm sorry, what did you say at the end?
That if you could please elaborate a little bit about the possible role of mechanics.
Yes, indeed. So that's part of the reading material that's been circulated. So I think what one needs to look at least in some cases it appears as if a Turing mechanism might work. As I showed you in the polyspondylium example, the pattern develops in situ without any significant rearrangement of cellular positions. But in other situations, there is significant cell movement, and whenever that happens, you have to take into account the role of the cytoskeleton, the role of intracellular movement of things, especially of these pigment granules, and of course of other forms of signaling between cells. Yes, based on mechanics, I mean. Yeah, I think Shigeru is going to go into this to some extent, so I won't comment further.
Okay.
But let me very quickly finish off my next 2 or 3 slides, shall I? Then we can.
Absolutely. Please go ahead.
Thanks. Right. No, so these are very general thoughts which I wish to place before you. See, one of the things that Turing said privately he wanted to do when he developed this model was to defeat the so-called argument from design. Now, this argument from design is a kind of old bugbear which has haunted biology, and this is the notion still held by the general public, let's say. This is the notion that if something is extremely complex, as living creatures are, they could not have come into being spontaneously, but rather had to depend on what you might call directed inputs. In this famous phrase made popular by Richard Dawkins, if you have a watch, not only did it have to be made by a watchmaker, but the watchmaker could not be blind. The entire approach of evolutionary thinking following Darwin and Wallace has been, on the other hand, that the watchmaker is blind. Therefore, as a questioner said a moment ago, therefore you can get non-random organization, in fact a high degree of organization, without putting anything particular in. Interestingly, the same attitude, the attitude that you did not have to put anything in, but nonetheless could get order based on what you might call natural law, was developed by an entirely different set of people who, curiously, were strongly anti-selectionist. Sometimes they're also called anti-Darwinian, but that's a tricky phrase to use because Darwin really thought of a great many things in addition to natural selection. But these people were certainly anti-selectionist. And sometimes this approach to form and pattern is called a physicalist approach, in contrast with the selectionist approach of pioneered by Darwin and Wallace. This physicalist approach can be traced back to Lamarck. By the way, I put Lamarck and Darwin Wallace here in quotes to say that I'm not necessarily referring directly to something they said, but I'm referring to them as the bearers of similar ideas. The physicalist approach is ahistorical, of course, whereas the Darwinian approach is very, very historical. The two approaches deal in different currencies. In the physical approach, you have, standard physical principles, the laws of mechanics, Newtonian laws, perhaps the laws of chemistry, all of which lead to self-organization. The Darwinian approach has to do with things which are not immediately obvious, not heredity by itself, which is understandable. That is clearly something all of us can see with our eyes, but with something called heritability, which is a measure of the extent to which heredity is important. And then, of course, this whole conception of fitness. Interestingly, both of these approaches are related to genetics or involve genetics. The sort of genetics that's involved historically was called differently. On the one hand, developmental genetics, which is quite recent, if you wish, perhaps 50 or 60 years old. And then transmission genetics, which goes back all the way to Mendel. To some extent, the dichotomies between these two approaches reflect a linguistic dichotomy between developmental genes and the genes involved in transmission of hereditary traits. And the dichotomy is in our minds, or was in our minds. It took a long time for people to realize that these are the same genes doing the same kinds of things, but in two very, very different contexts. I should also say that Lamarck is particularly relevant to this meeting because his ideas, which were based on physical principles, strongly depended on fluids, pressures, mechanical factors like elasticity, and so forth. Unfortunately, Lamarck has been caricatured a great deal in the literature for some of the things he said which were wrong. And one should go back as far as possible to the original Lamarck. I understand there's a scientific biography of Lamarck coming out soon from Stefan Tirrar, that we should look forward to. In any case, there's a movement on now in evolutionary biology. All of you will have heard the name by which it goes. Sometimes it's called Evo-Divo. Sometimes it's called Eco-Evo-Divo. All of these are short forms for saying that there are ways in which you can use the physical approach to form and pattern, which is ahistorical, but make use of variant outcomes resulting from such an approach. And these variant outcomes go by the name of phenotypic plasticity. Make use of these variant outcomes to now not do anything illegal, but to show how these outcomes could act as a substrate on which natural selection could act subsequently to firm up the outcome. So there's a very interesting way of saying that you could have a change that occurs on account of physical principles and then gets, Waddington's word was assimilated. Baldwin came up with a different mechanism. But this change which has occurred from physical principles has then become heritable through a mechanism which we will not look into now, but which is perfectly conventional. Nothing mysterious about it at all, and has been demonstrated to work experimentally. So that's one point that I want to leave you with, which is that even though, by the way, okay, it's good I remembered this in time. See, Turing, apart from wanting to defeat the argument from design, also wanted, like Darcy Thompson, who we saw very briefly, Turing also wanted to show that you did not need natural selection to explain many features of form. I'm not sure whether Turing was strongly anti-selectionist. Darcy Thompson certainly was. He thought that natural selection went too far. But both of them, like Darwin and Wallace, tried to give entirely materialist explanations for living form. Worth keeping in mind, even though they went on parallel tracks. Good. And here again, just to set the historical record straight, people speak a lot of, well, use the word epigenetics a lot and speak of Waddington's epigenetic landscapes. It's interesting that the idea was really thought of by another distinguished biologist, Sewall Wright, much before Waddington. And what's curious is that just as Waddington did, Wright also drew epigenetic landscapes. You see one of these landscapes here. So basically the idea is to say that the development of a fertilized embryo can go in various pathways, and the precise path which a particular cell chooses depends both on external and internal physical interactions, as well as on genes and gene products. Really very much the symbol that Waddington used. It's curious, however, that Wright's contribution to this idea is not acknowledged. I suspect part of the reason is that this diagram of his really does not look like a landscape at all. In fact, you have to study his paper and go through the diagram in detail to see what it's like. Whereas Waddington had the brilliant idea of asking an artist to depict the landscape for him. So it worked, right? So this sort of marrying of physical inputs or physical insights with evolutionary change has been attempted in recent years in all sorts of fascinating ways. Remember, I showed you this Newman-Frisch model for limb patterning. Well, Zhu et al. Have gone a bit further and have shown within a Turing kind of scheme now that you can, within a reaction-diffusion scheme, that may be more accurate, that you can get not only good mimics of the four-limb skeletal pattern in this character called Tiktaalik, one of our ancestors, certainly one of the earliest tetrapods. Not only can you mimic the pattern in Tiktaalik, but also in other creatures who are not directly ancestral to Tiktaalik, or at least we can't say, but clearly showed rudiments of pattern which are not seen in any developing limb. So the point here being, you can mimic evolutionary change by using Turing type mechanisms. That doesn't of course prove that that's the way they work, but if you like to think along evo-devo lines, it certainly gives room for thinking that this sort of mechanism, with all the uncertainty in it, remember the Bard and Lauder showing that it, for example, did not show scale invariance. So this sort of mechanism with all the uncertainty in it was later firmed up via genetics and natural selection. But that's just for thinking. And I end with this last slide. Turing's mother records that as a young boy, one of the things that he enjoyed doing the most was just sitting in the garden and watching daisies grow. Of course, daisies are one of the patterns that we tried to mimic in his model as well. So it's nice to keep staring at them as we think of Turing and what insights he has given us. The book there is a biography by Alan Hodges, which is well worth reading. I'm finished there. Thank you.
Thanks again for this nice talk. I wonder if there are more questions for the speaker. I don't see anything on the chat. If there is not, I would like myself again to raise a question for you. It's about the robustness of these patterns. So, I mean, many times in biological systems, the fluctuations can actually be pretty large, the so-called noise. So I wonder how nature, actually, what are the mechanisms, if there are any, that you can imagine that nature can actually use for actually produce robust patterns, Turing in this case. Thank you.
Yeah, see, the Turing mechanism by itself does not produce very robust patterns, as I told you. In other words, if you change the boundary conditions drastically, if you change the chemistry in some significant way, the Turing mechanism fails to reproduce the same pattern. So it may be, this is my thinking, this is not by any means proven, it may be that the sort of speculation that I left you with earlier is what has happened during evolution, that a Turing sort of mechanism, or for that matter, any other purely physicalist mechanism, has given rise to a pattern that has been selectively advantageous for whatever reason. So let's say for the zebra, by the way, this is hotly disputed, but let us say for the zebra, having stripes enables it to camouflage itself in its environment. The argument is that if a selectively advantageous pattern is produced for whatever reason, then one of these other mechanisms, ultimately based on genetics, can kick in to stabilize the pattern. So that you have specific regulatory genes set up now, which make sure that in spite of, let's say, temperature variations, in spite of different levels of nutrition, in spite of growth not being as full, as complete as might have been expected, feedbacks due to other genes help in and stabilize the pattern. But these cannot be thought to be a consequence of the Turing mechanism. There must be secondary consequences which have arisen in evolution precisely to act as agents of stabilization. By the way, notice that this also recalls to mind something we keep forgetting often, which is that natural selection does two rather different kinds of things. One, it succeeds in a certain mean result being achieved. And the second thing is it succeeds in narrowing the variance around that mean result. And these are two different consequences of gene action via natural selection.
Okay, thanks. I would like also to encourage the people attending the chat to visit the link that Professor Kondo put in the chat in order to actually play a little bit with simulations. That being said, I do have a lot of questions myself, and I believe that some of them are going to be addressed by Professor Kondo. But in any case, I would like to raise another question here. That is the following, is that what is actually the role of dimensionality? Because some of the samples, or most of the samples that we have seen in the talk today, have been addressed in the context of 2D, 2 dimensions. But actually, what do we know at this point about the role of dimensionality and the possibility of having 3D Turing patterns during development?
I can only answer this partly. I'll request Shigeru to correct me and also amplify on what I say. 3D patterns have not been studied as much for two reasons. One is, as I said, in some cases, it appears that you're dealing with quasi one-dimensional situations. In other cases, for example, the surface of an embryo, you're dealing quite a reasonable approximation with a 2D situation. So a real full-fledged 3D treatment does not appear to be required in many cases. And on top of that, of course, you have the fact that solving equations in 3 dimensions when you do it on a computer can be quite an intensive task. There's a person called Axel Hunding who began this many years ago to study Turing patterns in 3 dimensions. He did get interesting differences, by the way, from the 2D situation. And they're similar to the differences you see between solving the Turing system in a square versus solving the Turing system in a long and thin rectangle. You can get things like spots becoming stripes and so forth. But I do not recall his coming up with anything qualitatively different. But I'll let Shigeru answer this better.
Maybe the last part of my talk is answering that question.
Okay, so maybe we can wait about that. I do believe that we have another question. So the question is by Sundar. So please, Sundar, go ahead and ask your question.
Hi, thanks a lot for taking us through the history of Turing patterns. Also, I was wondering more about how you ended the talk with respect to selection of Turing-like patterns through evolution. So I'm wondering more like activator-inhibitor systems are actually quite common in developmental biology, right? So, but in fact, only very few cases of Turing-like systems have been really experimentally demonstrated. For example, digit patterning via the BMP SOX Wnt molecules that was shown in 2009-10 by James Sharp lab. So one reason could be that for this relatively very few cases being experimentally demonstrated could be the difficulty in addressing Turing-like systems from a molecular perspective, addressing diffusion in a real biological system and so on. Could there be an alternative hypothesis where biological systems are actually good in actively suppressing Turing-like patterns? Because given that you have a lot of activator-inhibitor systems in developmental biology, why are we not seeing more of them, being addressed in a way, right? or are we not visualizing these in a, are we missing something in visualizing these better?
I'm not sure what the question means. Are you saying why are there in fact not more Turing systems seen in biology, or are you saying why have people not worked on more Turing systems?
The former. So why are we not seeing more Turing-like systems given the prevalence of activator-inhibitor sort of interactions in developmental biology?
No, one second. If you mean in terms of identifying activator and inhibitor molecules chemically,
yeah,
I would agree with you. As far as I know, other than the example you mentioned and probably a couple more that we will hear from Shigeru, there may not be too many. But in terms of patterns which plausibly look like Turing patterns that can be experimented with at the level of pattern, which look like Turing patterns, I think there were a great many. So stripe patterns, in a great many systems are one example. So in fact, I would say that you get them in many places. But please remember, if you have an extremely mosaic system, so for example, if you look at some marine invertebrates, like let's say Ascidian, like tunicates, right? It may well be that the egg is so partitioned already at the beginning that you just don't have any scope for a Turing kind of mechanism to develop. The pathway of development is predetermined by cytoplasmic factors to begin with.
Yeah.
So it really depends on which system you're looking at. So here we are looking at systems which show mosaicism at an early stage. Mammalian development shows that, certainly. Many of these simpler systems show that, like Hydra, like the slime mold. They show regulative development. And as I said, there are cases like coat color patterns, which seem to be peculiarly suited to Turing type mechanisms.
So may I answer from the experimental side?
Yeah.
To the question? Yeah, the reason why only activator-inhibitor mechanisms are found in the real system is the researcher want to find activator and inhibitor. That's the reason. Because the activator-inhibitor scheme is very easy to be understood, even for the experimental biologist. They want to find the molecule, activator and inhibitor. But of course, many other related similar systems, something like the mechanochemical or the neural network work, and neural net system with short local activation and long inhibition can produce the same pattern. But that finding a molecule is much more easier than other more complex systems. This is a simple reason why the only activator and inhibitor system is found in the real system. This is my understanding.
Yeah, thanks a lot. Yeah.
Okay.
However, I would like to add on that regard, again, the question of dimensionality, right? Well, it's not really a spatial dimensionality, but in terms of the genes that could be involved in a particular interaction. Some of the works and most of the works that we have been introduced here about the Turing mechanism are dealing with two species, but actually you may have more species than that maybe their interaction in terms of activation or inhibition is not evident when you are looking at these two molecules, but actually only when you are looking at the whole network of interactions, right? So many times I believe that it's kind of tricky eventually calling something an activator and inhibitor when you are taking into account the whole number of interactions that could be involving species. Maybe I am wrong, I don't know. So if there are no more questions or comments, I would like to thank the speaker, and I guess that we will resume in more or less in 1 hour at 4:30 India time, and we will then listen to the talk by Professor Konda. And remember that in Slack, you can continue the discussions that we have here during the talks. So thank you so much, and we'll be seeing you in more or less one hour. Thank you.
Javier?
Yeah.
Just a
Thank
small,
you.
I think Professor Konda wanted to introduce the software. So people can play with that in the break.
Okay. Please go ahead, Professor Konda.
Yeah. So please close your.
Vidya, you'll have to unshare. You'll have to stop sharing.
Please shut your the slide, Professor Nanjundiah. Yeah. Okay. So I, at the end of the first part of this lecture, I just want to show my software to experience the Turing pattern. So because it is very difficult to get the concept of Turing pattern or feel Turing pattern because it is the outcome of the very complex nonlinear partial differential equation. So I made a simple Turing simulator software that can run on your web browser. So just this is a random clear point. You can change the initial condition and then calculate. And that is making the stripe pattern. And also you you can start with a blank and you can write your initial pattern and then run. Okay, again, we can get the Turing pattern. And of course, we can disturb the pattern and run. It can self-regenerate. And also, you can change the pattern by changing the parameter value. Oh, this is an equation. It's somehow complicated, but you have the preset, the parameter set. This is a stripe, so this is making the stripe. But now this is spot, reset, then from random, now you have spot pattern. And the network reset, you have a reverse spot pattern. This is called as, usually called as network. Or sometimes this is some giraffe pattern. Maybe it needs some initial pattern like this. Okay, then we have giraffe pattern, and maybe we can make more dynamic pattern, something like this. This is just a chaotic pattern that never stop, like this. Or this is dividing spot, like this. Ah, no, I'm sorry. Then go. Why it does not divide you? I'm sorry. Or stop. This also can produce the Berezov-Jabotinsky wave like this. So but just by changing the parameter value, it is possible to make this variety of stable and dynamic patterns. This is one of the patterns I like. Okay, even even from just a random pattern, it will making some oscillating spot pattern, and it is dividing and filling the field. Okay, so maybe now's the time to take the break. And so when do I need to come back here?
So 1 hour?
Yeah, in 1 hour.
1 hour. It's okay. But maybe my part will need more than 90 minutes. So I'd like to start sometime before.
That's fine. So, can we resume in 45 minutes? 45 minutes then?
So, okay. 50 minutes break is okay?
Yeah, perfectly fine.
Okay.
Yeah. 15, you said?
15 or
50.
50, okay.
50.
Okay,
So
thank you so much for the software. It was very nice to see.
Okay, please enjoy. And this is very important to feel what is the Turing pattern. So it is quite dynamic, and it can change by themselves. And this is the property. This is a very important property for understanding my part of the lecture.
Okay. So thank you, everybody, and we will resume at 4:20.
4:20.
Okay. Thank you so much, everybody.
So,
See you there.
Okay. Some people say that they. The audience are able to play the software?
Yeah, I have been able to. It is working.
Yes.
But someone said they can't. So
I don't know. It's actually working for me too. I guess that it's working for them, for most people. So, yeah.
Okay.
Okay. Thank you so much. See you in a bit.
Yeah.